inside the membrane of the neuron, and you measure the potential relative to the outside, okay, what you want to measure is minus 65 millivolts. So these cells maintain a hyperpolarized means more negative, it's negative, okay? So basically, hyperpolarized is more negative than resting. Resting is minus 65 millivolts, so most neuronal cells have a resting membrane potential of minus 65 millivolts, right? So basically what you have is a separation of charge across the membrane of your neuron, okay? So it's negative inside, right? So this is a zoom of this piece of membrane right here, okay? So you can see that there's a separation of charge, more negative with respect to the outside by about minus 65 millivolts, all right? So the maintenance of this resting potential is actually important. It's important for the cell to have this particular resting potential in order for it to be able to generate an action potential and transmit electrical signals. Okay? So the maintenance of the generation of this resting potential, which is this charge separation across pretty much all cells, right, is done via ion channels. Now I'm not going to talk to you about how ion channels establish the resting potential, but I'll tell you is how they generate an action potential. So basically an action potential is a change in the distribution of this charge. And it's a change that propagates along the membrane of your axon. So you start, let's say, here, right? And you disturb this balance, for instance, making this more positive inside by opening the channel and allowing an ion to move to the other side of the membrane. That's sufficient to actually make a change in the transmembrane potential in that particular part of your neuron. All right? So an action potential is a change in the distribution of charges. So this is a transient change in membrane potential, which is due to a change in the permeability to ions, right? How do you actually change this distribution of ions? You have to make the membrane more permeable to a certain type of ion. Ions are charged, right? We know that. So ions are positive, chloride ions are negative, right? If you have a net flux of one of these ions across the membrane, you're going to change the membrane potential. So, and that is going to change the permeability of your membrane to that particular ion. So the way you change the permeability is to open a path through which the ions are going through, right, in the membrane. And that happens via ion channels. So you change the state of these ion channels from being closed to ions to being open to ions, right? In the resting state, the ion channels are closed, no ions are moving. In the open, right, when you have an action potential, ion channels open, ions move, and you change the state of the charge distribution. So, ion channels. So, what are ion channels? This is the membrane of a cell. This is the lipid bilayer. You guys know that the membranes of the cells are made out of lipid bilayers, right? These are the phospholipids, and these are the hydrophobic tails, right? So they're head to head. So this is the outside of your cell, this is the inside of your cell. And here's an ion channel. So basically an ion channel is a transmembrane protein that creates a pore in your membrane, an aqueous pore, that allows ions to diffuse across. Now why don't ions diffuse across membranes of cells? It's because ions are charged, right? They're charged entities. They don't like hydrophobic environments. So it's actually very difficult, very costly energetically for a charge who likes clean water to go into hydrophobic environments, right? So what you do is you make something like this, make a pore in the membrane that allows ions to go through. And you want these pores to be gated because you don't want them to be open all the time, you don't want them to be closed all the time. You want them to open and close when it's necessary, right, to perform functions. So basically, ion channels are not just simple pores, massive holes like I just told you. They're actually have pores that select for a desired ion. They have gates here that open and close, and they have domains that modulate these gates, right? Because you have to gate in response to a signal, right? You have to open and close responsibly. There's some sort of intercellular messenger that binds to a domain of your protein, and this binding says to the ion channel, well, now you have to open your gates because ion I need to generate an action potential, right? So this is what happens. So you have to have these gates that actually are modulated by gates. So basically, when these gates are closed, you cannot have ions moving. But when the gates are open, so I'm showing here potassium ions because I'm working with potassium channels. So when the gates of the channels are open and you have some sort of driving force, right? You have to have a driving force. Because these are passive holes. You open the gate, ions can go through, but they will only go through if there's a driving force. For instance, a concentration gradient or some voltage that's applied across the membrane, right? These are charges. Now what happens when that occurs? When you have a gradient, you have a unidirectional flux of charges across the membrane, and that creates electrical current, right? Charge moving in time creates an electrical current that you can actually measure. So basically, what this is here is a single channel recording. So this is one of the first assays that recorded single molecules, okay? That recorded the activity of single molecules. And it's an electrical assay. It can record electrical current flowing through single ion molecules. So what this is plotting current in picoamps, very small current, right? You needed a very device with a very high signal to noise ratio, a good amplifier, and it took a while until people got this going. And it's recording current as a function of time. So when the channel is closed, your current is zero. This is the zero current level. When the channel opens, you get an instantaneous transition to the open current level, which is whatever picoamp, value. Let's say it's 2 picoamps. So this current is basically a single channel current. So you can see openings and closings, right? You can see the breathing of the single molecule, right, in real time, because you're recording it. Of course, there's a certain resolution, so it's only as good. The time resolution is only as good as the time resolution of your detection instrument. So this 2 picoamp value basically tells you how many ions flow per second in this through this open port, this single port of this single molecular machine. All right, I'm done with this one. Now let me tell you how the action potential gets generated. So why did I tell you about this? Why are ion channels useful? Basically, ion channels are expressed on every single one of it, in every single one of your cells. They're expressed from bacteria to humans. Okay? You have tons. There are tons of types of ion channels. There are selected for all sorts of different ions, and they're activated by various stimuli. So one of the most famous roles of ion channels is to help in the action potential generation that I just spoke about. So here's the membrane of the cell. Here's, again, the lipid bilayer, outside, inside. And there are gradients of your physiological solutions, right, that are set up so that there's a huge gradient of sodium ions from the outside to inside. Right? So you have high concentration of sodium ions outside and very high concentration of potassium ions inside. So you have inverse gradients of sodium and potassium. And that's very important for the generation of these electrical impulses, for you to be able to function. So you have to maintain these gradients. And those are maintained by sodium-potassium pumps and all sorts of other proteins, calcium pumps. Depends on what gradients, what ion gradients are actually maintained. But in axons, in neurons, you have sodium and potassium, which are the main players. So here there are 2 channels in the membranes of your neurons. Here's a sodium channel in red and a potassium channel in gray. So normally, the resting potential I told you was about -65 millivolts. So here's what I'm plotting here is a change in voltage in membrane potential as a function of time. So you start at -65 millivolts, and then there's some sort of stimulus that comes across that depolarizes, and when I say depolarizes, makes the inside of the membrane a little bit more positive than -65. So when I'm showing here a little bit of plus, that's just a little bit more positive than -65. So you're depolarizing slightly the membrane. Now these channels are very voltage dependent. They're very sensitive to voltage, but in different ways, right? So sodium channels respond very fast to changes in voltage. So what happens is they sense the voltage and they open their gates. And now what's going to happen to the sodium? It's going to go in, right? charges, right? Because you only have a certain type of ion moving, so it leaves the chloride ions behind, because your solutions are electroneutral on both sides, right? You can charge solutions. So you're moving across the membrane of positive charges, so you're making this inside more positive, right? Making this more and more positive. So here's what I'm plotting here. This gets getting more and more depolarized, right? And now, after these channels open, they inactivate very fast. And then potassium channels open. Now potassium ions are going to go the other direction, right? They're going to start going the other direction. So they're taking all this positive charge that you've accumulated due to the sodium channels opening, they're taking it all back out, and you're repolarizing your membrane back to its original value. Okay? So this is your action potential. So the reason I'm showing you this is for you to see how important it is that these two channels are actually selective for those particular ions, that you want to only have sodium ions going through here, potassium ions going through here, and you want them to open and close at the right times to actually give you this pulse, this action potential. Yes?
So is there any relation to this in taking like multivitamins and getting vitamins to make the energy your body has for eating properly?
It's way too far. It's not, no. The dietary, instant dietary effects are not reflected in the shape of your action potentials. Yeah.
So the stimulus changes the potential of the membrane, but in the one channel, like a gray one, it's closed.
It's not just one. There's a lot.
Oh no, actually, so for me, it's the pinpoint, right?
Yeah.
That's the gray one.
Yes. So basically, one of them is more responsive to changes in voltage than the other one. So voltage-gated potassium channels open slower. They don't have such fast kinetics of opening, and they take more depolarized voltages to open. So this one, let's say, opens when the membrane potential reaches -60 millivolts, let's say. Just difference, I don't know exactly what number there. And this one opens when it reaches -40, let's say, right? So this one opens first, it does its job, it inactivates, and then it makes it possible so that this one opens, right? Because this one opens, allows positive charge to come in, polarizes it further to the level where this one's in. So the timing is important.
Like so almost different channels have different sensitivity to.
Yes, yes, yeah.
What if you don't have the similar potassium?
Oh, you do. You do. There's the physiological solutions are 150 millimolar sodium outside and about 100 millimolar potassium inside, and they are very large sinks. Okay, so you have very large volumes of your body fluids, right? Blood, a lot of that, and your channels are very tiny, and you only need a few ions to move. The channels are a nanoscale. Okay, so you only need a few ions to move to actually locally polarize the membrane, and that particular change in membrane potential gets propagated further and further down the axis. Okay, so you just need a few ions moving to generate an action potential. Yes. Oh, sorry.
When at the end there's more potassium outside, and at the beginning there was more potassium inside, are there other channels that reestablish that, or does the body just count on there being enough ions in solution that it's not significant?
So this is a complicated. Your solutions are always going to be electroneutral. Your solutions are never going to have more, right? You're not going to have positive solutions or negative solutions. Your solutions are going to be always electroneutral. So as I said, your separation of charge occurs very locally across the membrane. So you're going to have, let's say, a couple of positive charges here and a couple of negative charges here. But overall, so that's a very small, that's a very, very tiny area of space. Okay? So this does not disturb the electroneutrality of your solutions. And you always are going to have enough ions. There's no problem.
How does the body do the crosstalk? Is it so close together?
How How does the beta-glucan what?
Crosstalk.
Crosstalk?
Two channels are nanometers apart, and you have suppose like one or two, right? I have one or two. Is there a chance of them just not serving up?
Right, so if they would be attached to each other, yes, they would probably disturb one another. Let's say that one of them moves a domain, right, and it just directly pushes on the other one. Is that what you're talking about? No, generally the motions are fairly small. The channels are not really in complexes together. These proteins are very tiny. They are aggregated at particular nodes. So in biology, I mean, in different cell types, you're going to have, for instance, in axons, there are certain nodes, things called nodes of Rambier, where channels are, for instance, sodium channels are very heavily distributed at nodes of Rambier, not so much in the other parts of the axon. So you don't have crosstalk unless you need to have crosstalk. Yes. Yeah.
How does the cell maintain the concentration gradients for the potassium and sodium ions individually? I mean, electrically, you can stay neutral, but still you need to have also the concentration gradients.
Right, so you have very large solutions, as I said. You have very large sinks. So your blood, your plasma, there's a lot of solution, right? Your concentrations, you have large sinks. And you only move a few ions every time you generate an action potential.
But still this goes on for a while.
And
So
there
it
are,
takes some-
yes, so there are sodium potassium pumps that actually work to reestablish gradients. So they move 3 sodiums out, 2 potassiums in for each ATP molecule. So there are 8 dependent pumps that do that. But even if you poison the pumps, you can still generate action potentials for hours. Even if you take a neuron out of your cell, out of your body, take a neuron in vitro and poison guavin, let's say, your sodium potassium pump, then you still can generate action potentials for a really long time because you don't need to move that many ions to change the distribution and to generate action potentials.
So in the case of electrotherapy, when they actually give electrical stimulus, including epilepsy and things like that, how does that affect this?
Oh,
Or
that's
is it
very
related to
complex.
this, or-
Yes, it could affect it, but nobody knows exactly how that works. It's very hard to measure these things. Yeah. But yes, definitely electrotherapy is going to mess with cells. Unlikely. Yeah. There are some other issues, and yeah, there's renal issues. I mean, your body's there's a lot of organs that your potassium deficiency would be more relevant first.
One question. Are the Chinese available? Are the channels available, or you have to introduce it into the network?
Oh no, the channels are made before when you start becoming a fetus. So that all these proteins are being made during development, and then we have different types of channels that are expressed during certain stages of development. And, whatever complement you have, and then I'm sure they change with age. All right, let's move on. Otherwise, I'm not gonna get past slide number 10.
It's Memorial Day.
Which is fine. It's Memorial Day, that's true. I have like, what, 20 minutes left?
Take your time, it's no problem. It's important that we have many questions.
All right. Good questions, guys. All right, so what is research in my lab? What do I do? Okay, so I just work with these ion channels, basically, and I work with this at the single molecule level. Basically, I'm looking to see how different domains of these ion channel proteins, these little devices, function, both separately and together, right? Because you have to have different modules in every one of these proteins. One of them gates, one of them is the core that allows ions to go through, right? And how do they function separately and together to allow these channels to fulfill these biological roles that I just talked about? bind, and what is the chemistry of binding, right? I told you, let's say you have a channel that is responsive to cyclic AMP, which is a secondary message. Cyclic AMP has to bind to your gate or somewhere near the gate. Then some conformational changes will occur, and then you have to transmit that motion to the pore, right? So that it opens the pore. So how does this work? How does the binding of the ligand open the channels, right? And which is what I'm going to talk to you about today. How does the pore select for the appropriate ions, right? How do you know to just pass potassium versus chloride, or potassium versus sodium, or sodium versus potassium? How does the pore select for the right ion? So I'm not going to show you these cartoons anymore because luckily we have crystal structures, so we have pictures of these channels at the atomic resolution. These pictures were taken with X-ray crystallography. You guys know anything about X-ray crystallography? I'm going to tell you a little bit about it. But basically, we know for some of these proteins, we know their architecture, whether alpha helical or beta sheet or things like that, where the amino acids are, right? So ion channels are made of amino acids, right? There's like tons of little amino acids, one after the other, and whatever amino acid, the amino acid composition dictates what the architecture of the channel is going to look like. Do you guys know what amino acids are? All right. So basically, these are a few of the channels of whose structures have been solved by X-ray crystallography with atomic resolution. All right. So basically, this is what we do. We actually try to solve new channels structures with X-ray crystallography, or we try to look at how these channels function at the structural level and at the functional level, and make maybe some mutants, and then see how the function is perturbed, and then crystallize them and see how the structure is perturbed. Okay. So the techniques that are used to address mechanistic questions about ion channels are what I showed you before: functional studies of these channels using either Pascal electrophysiology or rigid bilayers. So basically, by looking at the functional, right, how they function, right, how they open and close their pores, whether they're still selective or not selective, things like that. So you can do that using function assays. We can do biochemical and biophysical studies using all sorts of other assays. We do X-ray crystallography of ion channels to look at their structures and how their structures have changed. And these other two are done in collaboration with other labs: electron microscopy of ion channels and molecular dynamic simulations. Basically, electron microscopy is just using radiation, and molecular dynamics is just basically theoretical calculations. This is not, doesn't involve experiments. You just take a file, do a protein, and say I have amino acids here, here, and here, and I'm calculating potentials and energies, and I'll show you. So potassium channels is what I work on. So potassium channels, this is a bird's eye view of a potassium channel from the top of the membrane. Okay? Potassium channels are tetramers, right? They're made of 4 subunits. And in eukaryotes, you can have different types of subunits from different genes. In prokaryotes in general, there's just one gene that makes 4 products. And these 4 proteins come together, okay? And they form the core in the middle. This is the core where the ions go through. And these are, structural domains or domains where things bind to change function. We don't know everything about them, right? So we're trying to figure it out. So potassium channels are tetramers, as I showed here. So basically you have 1, 2, 3, 4 subunits in different colors, right? Shown in the 4. They have potassium channels, which is very interesting. They have been conserved. They are conserved from bacteria to humans. Okay, they have the same architecture and similar architecture and similar properties. They are ubiquitous and they are a very big family. So they share a common core, but all the other domains are different, and they're tailored for the function that they're supposed to perform. Okay, so their channels that are activated by voltage, like the ones that I showed you earlier for the action potential. There are channels that are activated by ATP, some by calcium. Calcium is another very ubiquitous second messenger. These are activated by CPMP. And these are the new bacterial channels that have been identified recently and that have been very helpful in actually determining the structure of these things. Because eukaryotic channels, for practical reasons, they are hard to crystallize and hard to determine the structure of. Because you can make them in high amounts and things like that. But bacterial channels, you can make in very high amounts because you can actually grow them in bacteria. So bacteria has a doubling time of about 20 minutes. You can put bacteria in a flask, and in like a couple of hours, you have tons of them, right? And if you have a gene for your channel that you manage to transform the bacteria with, and you induce it to make protein, and it makes so many plugs, right? You can actually isolate a lot of protein from your bacteria. So it's a very good workhorse for biochemists, full life bacteria. So domains, you said? Right. So the channels, that's the reason I talk about domains is that functional domains. There are some parts of the protein that are responsible for binding your ligand. There are parts of the protein that are responsible to pass the ions through and things like that. So usually we separate proteins in functional domains because they're not just performing one function, they're actually doing certain things. And there's like Legos put together, right? So there's this domain that you can attach to this domain together, and you can have this function, right? And you can modulate it, or you can make an energy bill with financing, right? So a lot of proteins are made of interchange domain. You can interchange domains actually. So you can manufacture proteins that have a domain from one protein and a domain from another protein. Put them together and they work as a unit. So this is nice. They're actually separate and you can plug them in, plug and play. All right. So that's what I meant by functional. So potassium channels are very selective for potassium ions. And the protein region that's responsible for selectivity is completely conserved among family members and blinds the spore, right? So basically, you would imagine that, right? So there's the chemistry that allows a protein to select for a certain type of ion has to be in the region where it's important, right? In the pore, where the ions are going through. All right, so KCSA is the first prokaryotic potassium channel that was ever crystallized. People got the Nobel Prize for crystallizing this channel in 2003. This is the first structure of ion channel 4 that was solved eventually at 200 resolution. So you can, so here it is basically. This is the outside of the cell, just to orient you inside of the cell. We have the 4 subunits, right, in 4 different colors. Right? They come together around the central core, so the core is going through the middle. So you can imagine that around it you have lipid bilayers, right? So the membrane of the cell goes from here to here, right? So this is where your lipid bilayer is. And these transmembrane domains, so these alpha helical structures, are transmembrane domains. They're hydrophobic, and they really like to be surrounded by lipids. Okay, this is a hallmark of membrane proteins. You have these stretches of amino acids, stretches of hydrophobic amino acids that project their hydrophobic amino acid side chains into the lipid, right? So that lipid hydrophobic likes hydrophobic, right? So that you have anyway, so that these proteins are actually qualified as transmembrane. So each of these subunits has 2 transmembrane domains. You see these 2 long helices that span the membrane, and then you have this loop here that constrains the pore right in this region. Okay, right? So this is actually the selectivity filter, something called the selectivity filter, where the ions are discriminated against. So this portion of the pore says, okay, I'm going to to let potassium ion through, but I'm not going to let sodium ions or chloride ion through. This one is a very large area here in the middle that is hydrated and it's not very selective. And this, you see how these helices come together like a bundle? This is called a bundle crossing. Enough. And this forms the gate of the channel. So this is where the channel opens and closes. So this is the structure of a closed channel because this bundle is closed and it doesn't allow any ions to go through. Okay? When the channel opens, these things come apart. Okay? They open the gate and they allow the ions to go through. And all sorts of other things. So I was telling you that this selectivity filter chooses between potassium and sodium. So I'm just going to tell you a little bit about this, and then I'm going to go through fast to the rest of the talk, just so you know. So basically, the selectivity filter is this narrow region. So potassium ions, so all these salts, right? What happens when you put sodium chloride in water? You take a little bit of powder, sodium chloride, and put it in water. What happens? You dissociate sodium from chloride, right? So what happens? Each of your sodium ions is surrounded by a bunch of water molecules, and each of your chloride ions is surrounded by a bunch of water molecules. Now, in order to dehydrate back one of these ions, takes a lot of energy, and that's given by the dehydration energy. Calculate that, right? There are certain numbers for it, and it's huge. But each ion has a certain amount of dehydrated energy depending on the size and the charge, right? So, in order to actually take waters away from ions, it takes a lot of work, right? So now this selectivity filter is very narrow, and it doesn't allow ions to come in with their waters of hydration. So in order for ions to go from this side of the membrane to the other side of the membrane, they have to shed their waters away. So, and this happens in the selectivity filter. They shed the waters and they go through here and they come back up and they rehydrate. But I showed you that the potassium channels actually process these potassium ions very fast. You get pretty large currents through your single molecule. Okay? So that's given by a combination of the fact that you can have multiple ions in 4, and because the selectivity filter looks like this. The selectivity filter of potassium channels looks like this. Now what am I showing you? So this is basically a blow up of this, right? And I'm only showing 2 of the subunits, right? So you have 4 things coming together like this. These 2 that I'm showing right there, and then 2 more, 1 towards you and 1 to the back. But in order to see them better, I'm just showing this. So these things, this is the peptide backbone, right? The yellow stuff. So this is the protein here. The green stuff are the potassium ions, the green balls, right? Are the potassium ions in the selectivity. What's red? Red is oxygen. Okay, so this is blue is nitrogen, red is oxygen. Okay? So as you can see, the oxygens from the carbonyls of the peptide backbone are facing into the core. Okay? The oxygens from carbonyls is like C double bond O. These oxygens are dipoles. Okay? I mean, they're not dipoles. They are part of a dipole, right? Similar to the water dipole, right? So the water is oxygen and 2 hydrogens. And so you have your negative dipole is at the oxygen and your positive dipole is at the hydrogen. Similarly here, right, you have a C double bond O, and that forms a dipole. The oxygen is the negative part of the dipole, and the carbon is the positive carbon. So these dipoles, these oxygen dipoles, are surrounding a potassium ion in the selectivity filter. Right? You have 4, 1, 2, 3, 4 from 2 subunits, and you have 4 more from the other 2 subunits. So you have 8 oxygens from 8 partially charged, partially negative charged oxygen dipoles surrounding a potassium ion in the selectivity filter. So that's similar to what? What did I say about water? So you have, in solution, you have a similar arrangement around the potassium ion. You have water dipoles surrounding your potassium ion with the oxygen facing towards the potassium, you lose by dehydrating the potassium ion to get it in here is actually all compensated in the selectivity. Okay? It varies between ions. So different ions have different dehydration energy. So in order to evolve a channel for a particular type of ion, which is where I think you're getting at, is you could make these binding sites, right, for that ion mimic what the coordination distance and coordination number of ligands is in solution. It's hard to know because we don't have that many structures of ion channels. So everybody would love to have a structure for a sodium channel 4, right? We don't have that. There is one bacterial sodium channel that doesn't have a sodium channel, but nobody was able to crystallize it. So we don't know whether the principles are conserved to sodium channels or calcium channels for that matter. Chloride channels actually are a little bit different, and it's not very clear. So it's actually very difficult to solve these problems, and this is why there was a Nobel Prize awarded for this. Because it took forever for people to figure out what is the selectivity principle. Yeah.
So on this model for the potassium channel, is there any reason why the sodium ions will not make it through? Because the sodium ion are small.
See, I got you where I needed you. This is great. So now, see, this is the question. So this is great for potassium ion, but what makes it so bad for sodium, right? Why is it that this thing rejects sodium? Very good. I like that. Must have done my job. And I have 5 more minutes. All right. So basically, this is very good for potassium ions. So you're going to see in a second. So what makes it bad for sodium? And the answer is, it's hard to know, but the hypothesis that was in the field a couple of years ago, 3 years ago, was so basically that this channel was crystallized with potassium ions inside. So you can basically see these densities, okay, with X-rays, right? You have a potassium channel with potassium crystallizing potassium. You get the structure and you can see densities for potassium. So it's fine, great. It looks wonderful. Now with sodium it's a little harder because sodium has the same number of electrons as water. Okay, 1. Another issue is that when you take potassium away from potassium channels, especially this ones, they change the conformation of the selectivity filter. So now you no longer have the same conformation. Now you have some sort of densities in the core. The resolution is not so good for some reason. You have a different conformation, and sodium ions have the same number of electrons as water. So you don't know what's sodium and what's water. Okay? So the hypothesis is that, as I said, the potassium dehydration costs are compensated in a filter due to perfect carbon position. So that's why it likes potassium. Sodium, however, is smaller, right? And the carbonyls are too far away. The sodium is smaller, and as I said, the dehydration energy is different, and you actually have to have waters a little bit closer together to coordinate a sodium ion and a potassium ion. So sodium is smaller, carbonyls are too far away to coordinate these bindings. So when you get a sodium ion in here, this is drawn to scale. Okay? Get a sodium ion in here, it doesn't like it. This is not a good binding site for sodium. Okay? So energetically, this is unfavored. Okay? So it takes a lot of energy to dehydrate a sodium ion, and then you don't give it a proper binding site. It doesn't like it. Right? So the current hypothesis is that sodium binding the selectivity filter is an energetically unfavorable process. So that schematically, you can draw potassium in solution has this energy well right here, and potassium in the selectivity filter is a similar energy well. So it's isoenergetic for potassium to get in here, in here, and out. But for sodium, the energy state, there's a higher energy state in the selectivity filter, and it doesn't like to be in here, and it doesn't go in. So that's the hypothesis. Well, we found that that was not, this is not what we found when we looked how do KCSA channels select against cellular sodium? So we looked at the process, at the rejection process from the inside. So we asked, is it a thermodynamical issue or is it a kinetic issue? The question being, is it true that it's not favored, that there is no binding site in the selectivity filter for sodium? Or is it that sodium is prevented from getting into the selectivity filter by a huge energetic barrier, right? So something like, you like to be there, right? You like to be in this particular room, but the door is too heavy and it's very hard for you to open the door. But once you're in that room, you like to be in that room, right? So this is there's a difference between this and there's this crappy room that nobody wants to be in, right? The door is wide open, but you don't want to go there, right? So that's the difference, the thermodynamic versus kinetic. So we looked at it with single-channel liquid battery. We looked at it functionally, and we looked at potassium current and how it's modified by sodium, all right? Then we did some calculations, theoretically, to see where you have energy wells and where you have barriers, right? Because we have the structure of these things that might as well use it. And we did some X-ray crystallography, and we did it in lithium because lithium is basically a better sodium. So lithium is tinier, it's smaller, so it's much more unfavorable to dehydrate it. It costs much more to dehydrate it, and also it doesn't go through potassium channels, so it's a better property. So this is the setup, and I'm not going to go over it too much. So we looked at single channel recording and how did this lithium modify the potassium current. So just to get to it a little faster, we saw that there are two. I'm going to just go through the chase. So sodium and lithium have two distinct effects. They modify the potassium current in two different ways, right? In one, they have a fast block, so there's a very fast process where they modify the amplitude of the single channel current. So basically, sodium goes into a binding site and goes out very fast, all right? And then there's also another effect, so basically that's by you lower the single channel amplitude. And another one is slow, is a slow block. So we found two effects of the sodium and lithium at the functional level. So the conclusion from that was that there are two different binding sites where these ions bind to give you these two different effects. But so while single channel recording is very good at telling you that there are two different binding sites because you see two different things happening, it doesn't tell you where those binding sites are, right? So for that, we needed a different technique. So the first thing that we looked at is molecular dynamics. So we wanted to calculate, so this is done at UC Davis by these two guys. So basically we wanted to calculate along the axis of the core, given all the charges and all the, hydrophobicity, and we have all these lipids around and waters and everything. At every point in this axis, what is the likelihood for what is the free energy corresponding to each position of any ion across this, right? So you basically want to see an energetic landscape. Where is potassium channel favored to sit? Where does it have a barrier? How about sodium? So what we saw was the following. We saw that basically this plots the free energy in the selectivity filter as a function of the distance in the selectivity filter. So let's just look here because we don't have much time. So we wanted to see what happens in this area. So there's a potassium ion coming from here and here. A lift potassium is in red, sodium is in green, and lithium is in blue. So these are energy wells, meaning that this is favored, okay? Wherever you see a minimum, it likes to be there, okay? So potassium, there's a well right here between -6 and -5.5 right here, right where it was seen in the X-ray crystallography. So this is not surprising, and this is good. It validates the calculations, right? So potassium likes to be in. Now, is there a binding site for sodium or lithium? Remember, we wanted to know, is there a good binding site for sodium or lithium in the selectivity? And it turns out that there is, right? There are binding sites. There's a very nice well here and a very nice well here. And those are pretty decent in value. So it's very favored, it's good, negative free energy. But they're not in the same place as potassium. So this is the trick. So remember how it was that if you put a sodium ion in here, oh, it's too far away from the carbonyls, right? And it doesn't, the core can collapse to coordinate the sodium. Well, the binding site for sodium is here. See, in between the S3 and S4 binding sites. Right in the plane of these carbonyls. Instead of being in the cage coordinated by those 8, it's up a little further. And these things are actually closer, right? So if you actually, right, you have this part, the hypotenuse is longer than the other sides of the triangle, right? So the distance from here to the center of this binding site is larger than the distance from here to the center here, right? We agree on this? Okay, so if you put a sodium ion or a lithium ion in this spot, you can actually properly coordinate it with 4 oxygens from the carbonyls and with 2 waters, one that sits here and one that sits here. Okay? So, with theoretical calculations, we found that there are wells at these binding sites for sodium and lithium. So basically right here. So this is the spot for potassium, this is the one for sodium, and this is the one for lithium. So with X-ray crystallography, this is in order to resolve 2 objects, you need the 2 objects. In order to, right? So why do we need to solve the protein structures with X-ray crystallography to determine atomic resolution? It's because you need to give them a scatter light that is of the order of with the wavelength of the order of the distance that you want to look at, right? So those are the X-rays, right? Angstrom resolution. So the peptide bond in proteins is about 1.3 angstroms, so you need angstrom resolution, angstrom wavelength. So that's X-rays. So in contrast, the dimension of a cell, which is what you see so far, is about 10 microns, so you can use 5 microscopy methods. And this is, you guys know what X-rays are, right? I shouldn't go through it, all right? But I love these animations. Anyway, so what you do is you have an X-ray source, you make a crystal of your protein. You know how you make crystals of proteins? You purify proteins, and you set about a gazillion trials, and maybe one of them, you're going to get a crystal. So proteins come together and in order, 3-dimensional rays. So you have a crystal of protein, and you shine X-rays into a crystal, you diffract your photons. Your photons are incident upon this detector, whatever film or CCD detector or something. You measure the amplitude, measure the number of photons basically, and that's your diffraction data. We won't go over the phase problem. This is going to do some other time. And oops. So this is basically crystal diffraction data model, and that's your protein structure. Basically, we solved the structure of this of KCSA in lithium, and we inferred that the binding site for lithium is right here, where it was shown by electrodynamics. Basically, in the plane of this carbonyl surrounded by waters. All right. All right, so I'm not going to go over the, so basically, molecular dynamics calculated an energy well at that particular spot. We saw it with X-ray crystallography. We didn't really see it, we inferred it, but anyway, because it has 2 electrons, right? So we know that lithium has 2 electrons, and it's very hard to see it with X-ray crystallography because there's not much diffraction of 2 electrons. But we inferred it due to the position of water and the confirmation of the selectivity filter. So then we said, okay, if there is a binding site for lithium and sodium inside the selectivity filter, that means that it's favorable, but it can't get there because of an energy barrier. And we calculated the energy barrier using, again, electrodynamics. So basically, we found out that in order to move ions from here to here, it requires about 45 kcal per mole. And that's about how much you need to this situation. So sodium is not favored in selectivity filter. It's favored, but there are larger energy barriers for sodium to get where it wants to go. Okay? And so those are the conclusions. Intracellular sodium and lithium are excluded from the core by a mechanism of selective exclusion and not selective binding. The selection is not due to unfavorable binding because we think we found the binding sites. The binding sites are, the reason they weren't found before is because they're in a different place. They're not in the cage of those carbonyls, but in the plane. And the selection occurs because there's a large energy barrier, because this pore is a multi-ion pore, and it's optimized for potassium ions. Okay, it's optimized for potassium ions and not for sodium ions. And basically, what we need to do is we need to measure directly the binding, right? If you can measure directly, this would be uncontroversial. If you can measure directly the binding of sodium and lithium and get a KD, right, for these ions to the channel, that nobody can tell you anything about this, right? There are a few problems with this, that the channels don't like to be empty of ions, and in order to measure binding of something to something else, you first have to have something without anything bound, right? The binding affinities are believed to be low, so you need to do equilibrium measurements, and you need large concentration of proteins. And you need to do this in lipid environments and not in detergent, because you need to measure proper affinities. So we are addressing this by taking the channels out of detergent, because when you purify these channels, you have to keep them soluble. And you give them detergent to keep those transmembrane segments that are hydrophobic. Remember, if you put membrane proteins in water and salt solutions, they're going to precipitate. You have to give them something to keep those hydrophobic transmembrane segments happy, and that's either lipids or detergents. But with lipids, unfortunately, you can't have them, can't concentrate them to high amounts. You can't have them very soluble. So that's why you use detergents. However, we figured out that you can put them into nanodiscs. Okay. So what are nanodiscs? They're these apolipoproteins. So here's the apolipoprotein in blue. So there are these belts of protein, okay, that surround this lipid bilayer. This is lipid bilayer here. There's a bunch of lipids. This is the apolipoprotein again. And this is a cartoon of how you put a protein inside of these nanodiscs. Okay? So these are the lipids. This is the protein that's basically an HDL carrier. Okay? And so this is your protein in the middle. So what you have is now you have a native preparation of the protein. Because this is one of the problems. You need to have high concentrations of these proteins, but detergents are no good because they're not native, and then liposomes are no good because you can't concentrate them to high enough concentrations. So this is the perfect compromise. So that's you can't do binding assays in lipid vesicles because, right, so this is the lipid vesicle, these are your proteins, and you can only have access to this side of the membrane, right? This side is impossible to access. But in nanodiscs, you actually have access to both sides, and you can concentrate them to whatever you want. So we actually did this for KCSA. We put them into these nanodiscs. So this is a cartoon showing the KCSA channel, and we did electron microscopy to show that you do have these structures, right? You have the protein actually inside the nanodisc. And basically then we used equilibrium measurements like isothermal titration calorimetry to measure the binding of the ions to the channels. So basically the way this works is it measures the heat that's generated upon addition of the ligand to your protein. Okay? So you can calculate KDs and delta H's and delta S's and everything. So this is the preliminary experiment. So these are the data for calculating the KD for binding of potassium to an empty channel, to an empty KCSA. So we found like 158 micromolar. So what we're going to do is the same measurements with sodium and lithium and see what the binding is. So we didn't finish that. These are the acknowledgements. Near Thompson is the graduate student that just finished. These are just the X-ray team. They helped us solve the structure. This is
the proteins and use the proteins for this measurement, or you have some
Yes.
neuron extraction?
Yeah, you purify, so you make the protein in bacteria, and there are protocols to purify the protein away from the membranes of the cells. And the way you do that is you dump a lot of detergent onto the membranes. So basically, you break your cells, and then you put tons of detergent to extract the protein out of the membranes of the cells. So usually, these methods are a little problematic because you need to find the right detergent. Not every membrane protein is right in every kind of detergent. Detergents can be very to nature, right? Like SDS is very to nature. So you need to, people that do this a lot of crystallography or membrane proteins, they take a large of E. coli, they split it into like 20, right? Into 20 aliquots, and they try 20 different detergent extractions, 20 different detergents, just to make sure that to hope that they hit the right detergent that will keep their molecules, their proteins in a native form, sort of, right? And not and get it out of the cells at the same time.
So in this method, you still extracted the protein and then bind it to it. So you use the
Exactly.
detergent.
So you first use the detergent to take it out of the cells, and then you take the empty nanodiscs, right? Just the apolipoprotein protein with the lipids, and then you take your protein, you combine them together, and you dialyze the detergent away. And in the dialysis process, your protein actually gets inside the nanodiscs.
Is there any way to work with neuron, instead of, proteins, you just can measure?
You can always work with neurons. Unfortunately, with neurons, you have everything in there, all sorts of proteins in there, right? Or even you purify the protein from neurons.
The signal from the neuron, right?
You don't know what you're measuring, right? Because first of all, you have a cell, you have a certain number of proteins inside that cell. You have like tons of proteins inside any given cell at any given time. You don't know what protein you're looking at. You don't know what the salt concentration inside is. You just don't know anything. That's why when you want to actually figure out how each individual player works, you need to take it away from its native environment, isolate it, and see how that individual one works, right? I mean, obviously at some point, after you figure out how each player works individually, you want to put them together and see how they work together. But you can look at signals if you don't know where they're coming from in the cell, right?
The electric signal is from neuron, right?
I'm sorry, what?
The electrical signal that you showed is from neuron.
Nothing was from a neuron. Nothing. It was basically purified protein and put into artificial lipid bilayers, where you control the outside and the inside and everything. Everything is just that one protein, and everything else is controlled. I can even control what membranes they go to, what lipids they go to. Yeah. No, sorry.
Are there also selective times for magnesium and calcium ions? This is the first question. And the second question is, would you expect to have similar observations for magnesium and calcium because of similar ionic radii and similar energy barriers?
So the first question was, there are indeed calcium channels that are very selective for calcium that also allow sodium to go through, but they don't allow potassium. Magnesium has transporters. They're not passive diffusion. They're carriers basically. So you find magnesium transported very much slower processes. The second question was that?
Do you expect to, extend the similar analysis to both these ions because of similar radii and similar energy?
You mean between calcium and magnesium, or between calcium and sodium?
For example, potassium and calcium, because they're in the same.
So it seems like the selectivity filter of calcium channels is different. It won't be carbonyls, it won't be oxygens, but we don't really know. I think that for calcium, people expect to have a side chain of a negatively charged amino acid like glutamate or an aspartate or. And the selectivity filter of calcium channels does have, basically calcium channels are complicated eukaryotes. There's a long polypeptide that it has also four it basically is a tetramer, but made from one polypeptide, just one gene that makes four different subunits similar to the potassium channel. The potassium channel has one gene that forms this four individual proteins, but this one is just one long polypeptide. And the sequence is different between each four. So there's like a D and an E and a K and an A at the same. It's not complicated. They're not the same. So it's not clear what the selectivity will look like, and I don't expect it to be potassium now, especially since calcium is divalent. It's fairly size. Magnesium is very small. Calcium is about the same size as potassium.
Other questions?
Right. Right. So basically, the fast block is a very fast kinetics. Basically, the on rate of the blocker to the site is very fast, and the off rate is very fast. Right. So if that process is very fast kinetically, and your time resolution can't solve it, right? You will see a diminishing of the amplitude. Slow block, what it means is that the off rate is very slow. So it gets there, right? But then it sits there for a while, which you can actually measure, right? So yes, the energy well for the slow block is deeper than the energy well for the fast block. So that's why I thought that there were two different lines. Right, so I didn't show you the cavity calculations. So there were calculations that was not cavity. There were some very shallow energy wells in the cavity region at large, fairly non-selective. So there are shallow energy wells in the cavity, and there are deep energy wells in the selectivity. So that hence fast block. Yes.
Is there a chance that the structure of these protein gates will be different when they are within the lipid layer and when they're isolated?
You mean in the cell
In the
versus
cell itself,
in
yes.
artificial? Yes, there's always a chance. Which is why we're trying to actually put them in as native lipids as possible. So you can put them in E. coli lipids. You can take them out of the cell and put them back in E. coli lipids. So we're trying to mimic the environment without putting any extra stuff around. So we're trying to mimic the lipids, but not having any other proteins. And then you can add players one by one to try to mimic a response which is yes.
So what kind of diseases would any problems with ion channels affect? And also, are they genetically affected, or can they be affected with age? Like, can your ion channels go bad with age, or are they like from
Everything goes
birth?
back to age.
Yeah. Because a lot of neurological diseases, some of them are genetic, some of them are
Right. So basically, I mean, there are a lot of diseases that are caused by malfunctioning of ion channels. In general, if you actually have any screw-ups in the ion channels that cause the action potential, then you're pretty much dead, right? So those are actually important for the function the way they are. There are drugs that target calcium channels. For instance, in your heart, you have basically the action potential in the heart is a little bit different than the one in the neurons. It has a plateau that is caused by a calcium channel, and there are calcium. I'm sure you heard about calcium channel blockers that you take when you have heart conditions, so that they actually modulate the plateau size. There's I'm not sure if you've heard of the long QT syndromes, which is those are diseases. They're causing arrhythmia and sudden death, and those are caused by a lot of different genetic problems, different ion channels. There are a lot of ion channels that can lead to the heart. lot of channels that can be affected, cause disease, renal problems. So I don't know about age. I'm not aware of anything that deteriorates with age as far as channels are concerned that they give you a one more question.
So with respect to binding potassium and you're reporting something that other people haven't seen with more lipid, like the spherical lipid structures with your nanograms. Is
Right.
there a possibility that are you able to control for orientation of your nanograms? Do you know which way the channels are oriented when you make measurements?
But see, you don't have a problem with the nanodiscs. The nanodiscs are just, you have like a tire. It's like me having a tire around me, right? The tire is the apolipoprotein protein, and around me, let's say there are a bunch of lipids, because I need lipids around my waist. Okay, so you put me like this in a container, and you have access to this part and to that part. The only thing you don't have access to is my middle side.
Yes, exactly. So you have access to both sides.
Yeah,
Are you
so-
able to control for the top versus bottom?
No,
Is the
because
binding
they're all-
happening on the inside, not the outside?
No, they're all exposed to the same solution. I mean, you take me and you put me in water, right? Water accesses me everywhere except for the middle. So there's no way to do this. If you want to target just one side, then you want to put them in liposomes, right? Because then you want to hide one side from the outside, right? So you want to put them in spherical liposomes if you want to hide and not change one side. In nanodiscs, you change both sides equally.
Yeah, so my question, I guess I'm not phrasing it right, is that because you're accessing both sides, do you know which side that binding is happening? Is
No.
it possible that it's on the inside and not on the selectively filter side?
You can't control that in nanodiscs. There's absolutely no way to control that in nanodiscs. So in order to do this, you have to do liposomes. And yes, whenever you have access to both sides, you're not going to know which side is affected. True, true. But unfortunately, when you put channels or any kind of membrane protein into liposomes, you have this random orientation because you have no way to determine how you are oriented. And let's say you have 50-50, right? So you have access to 50% of your channels, but you think you have so many other channels, you have no control. That's my point with the liposomes. When you don't know how things are oriented, it's better to have equal access to both sides, right? If you have some sort of handle, then yes, that would be a nice orientation. You don't have it.
Okay, I would say let's continue our questions over the break because I want to have the last question. So is it correct to say that the transport is entirely due to diffusion? Transport across channel? This relates to my talk in an hour now.
Pretty much, yes. It's you basically open a pore. You open a pore, and there's a certain energetics inside the pore that is going to dictate how ions move to some extent, kinetically. But you do not get, the reason I said diffusion is because you don't get, there's no active transport, there's no carrier via a protein. You just open an axis, the pore, and ions are moving through. And they might be going faster or slower depending on whether you have negative charges, let's say at the mouth of the pore, positive charges at the other side of the pore, right? You would have a barrier, you have a little barrier on the inside or a smaller barrier to the outside, or they're attracted, right? So there's an energetic landscape that affects the movement, but the movement is via diffusion.
So in a live cell, let's say, does the ATP level affect the ion transport?
ATP level.
Yeah, in other words, does it need, do you need
No,
to
so-
fill the cell with energy? Or
You do not, that's what I'm saying. So there's nothing active about transport force. You just open a passage. And the movement is, as I said, dictated by electrostatics to some extent. But I mean, you could have a gate of the channel that is modulated by, right? Let's say your channel is closed and you in order to open that gate, you need to hydrolyze ATP or bind ATP.
That's what I was
To
wondering.
that extent, yes. But once you open that gate, the movement of the ions is
Okay. Well, thank you very much. Let's have more questions over the break.